In this video, we will discuss about cancer-associated fibroblast. So this cancer-associated fibroblast is a heterogeneous population that comprised of different kind of cells, which is pancreatic stellate cells, resident tissue fibroblast, adipocyte, bone marrow-derived macrophages, epithelial cells, mesothelial cells, mesenchymal stem cells, and they are mainly categorized into these three different cancer-associated fibroblasts. That is myofibroblast, inflammatory fibroblast, and antigen-presenting fibroblast. So this cancer-associated fibroblast, its major progenitor cells are these pancreatic stellate cells and the resident tissue fibroblast. So they are comprised of this. And the other cells that further joins are then differentiated into or converted into the cancer-associated fibroblast. So we will start our discussion with this myofibroblast, cancer-associated fibroblast. So one of the main characteristics of this myofibroblast is that they are close to the tumors, and at the same time they are high alpha smooth muscles. So this can be identified by checking the high smooth muscle signals. And on the other hand, this inflammatory cancer-associated fibroblasts are slightly far away from the tumors, at distant from the tumor, and they are low in alpha smooth muscle. They are low in alpha smooth muscles, and whereas this antigen presenting CAFs are also further away from the tumors, and the function of this three fibroblasts is to help in the tumor proliferations and at the same time helps in the immune suppressions. So what researchers has found out is that if they knock out these alpha smooth muscles in the myofibroblast, then it will make the cells more aggressive or proliferative or metastasis in nature, because now there is no barriers and the tumors can spread more aggressively. However, at the same time, cells become responded to the therapies, and one can target this pancreatic tumors with alpha smooth muscles removed or knocked out. This is one of the approach, that by knocking out alpha smooth muscles in myofibroblast, it has one advantage and one disadvantage. Advantage is that now they become sensitive to the therapeutic treatment. However, they become more aggressive and can metastasize or proliferate at different sites. And on the other hand, this inflammatory CAF, they have the ability to release cytokines such as interleukin-6 and LIF. I'm not able to recall the full form of LIF, but those two cytokines, they help in this tumor suppressions. So by targeting those cytokines can be one of the therapeutic approach to make the tumors less immunosuppressive. The other one, or the cancer-associated fibroblast, is this antigen presenting CAF. So this antigen presenting CAF, although it has the antigens that will activate the T cells, but this CAF does not have the costimulatory derived that can sustain or proliferate those T cells, and because of that it won't be able to generate immune response that are sustained, and in another way it will also help in the immune suppressions. So and this antigen presenting CAF are differentiated from mesothelial cells, and it is negatively related to the CD8 plus T cells by Treg ratio. That is, when the antigen presenting cells are antigen presenting CAF are high, it means the Tregs are also high. That will help in the immune suppressions. So by targeting these mesothelial cells, one can reduce the Tregs and increase the cytotoxic CD8 plus T cells, and thereby increase the immune efficiencies. So these mesothelial cells could be a potential therapeutic target to increase the therapeutic to increase the immune cell sufficiency or therapeutic efficiencies. And it can be clubbed or with anti-immune checkpoint blockade therapies or synergetic effect. So this is how the cancer-associated fibroblasts are categorized and how they can be used to improve the immune, to suppress the immune suppressions to improve the immune suppressions and increase the tumor suppression properties. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.